Screening expression libraries. Here we will start with expression cloning. If DNA library it is established by using expression vectors, then each represented clone will express and produce a polypeptide chain, and then it is further screened after expression. I think you are well aware about the expression vectors that are used to construct expression libraries. अक्सर औकात ये हो सकता है कि अगर हम टारगेट क्लोन को इंसर्ट करें होस्ट ऑर्गेनिज्म में बाय यूजिंग एप्रोप्रिएट क्लोनिंग वेक्टर, तो ज़रूरी नहीं है कि ऑल द टाइम ये क्लोन जो है वो होस्ट ऑर्गेनिज्म में एक्सप्रेस भी करे. अगर ये एक्सप्रेस भी करता है तो इसका जो एक्सप्रेशन है वो बड़ा लो या पुअर होता है. सो अंडर सच सर्कमस्टांसेस टू regulate the expression of the target clone, expression vectors they are used. So one can say that expression vector they can upregulate the expression of a target gene, especially in biotechnological applications. But keep in mind, this high expression, it is not always desirable for host microorganism. The reason is that if the target clone it express some hazardous gene, and then its expression it may be harmful to the host organism. So under such situation, it is also very important to regulate the expression of such genes. But overall, we can say that expression vector they can use to upregulate the expression of beneficial genes. So in case of expression vector, the main target is that to regulate the transcription level. So the control is transcriptional, so that we can produce high quantities of messenger RNA that will later on translated into functional protein product. Sometimes what happens that even native promoters of the target clone they do not express into the host organism, or if they are expressed, the expression is again very poor. The reason may be that RNA polymerase it is not having a high affinity in the host organism. So this situation it can be overcome if we use a strong promoter to express our target clone. Now number of strong promoters are available, and here if you remember I quoted the different examples like one can use LAC operon operator, TRIP operon operator, or even the hybrid of TRIP or LAC operon they can be used, and lambda promoter like PL it can be used as a strong promoter to upregulate the expression of target clone. Now we have constructed the expression libraries. Now how we can screen them, we are going to discuss different strategies. And this expression library construction, it is valuable when we may compare it with cDNA libraries or DNA libraries or by other PCR methods for the screening of such DNA or cDNA libraries. The reason is that here the screening it is not based on hybridization or by probe method, rather the screening it is based on the expression of the product. So the product it is expressed, and then different approaches can be employed for its screening that may be based on the structural and functional properties of the expressed product. तो यहाँ हम ये कह सकते हैं कि जो एक्सप्रेशन की बेस पे जो स्क्रीनिंग है इसका एडवांटेज ये है कि अगर जीन जो कि ख़ास प्रोटीन को एक्सप्रेस कर रही है उसका जीन सीक्वेंस अननोन है, तो अगर प्रोटीन प्रोडक्ट एक्सप्रेस हो गया है तो by using different type of techniques it can be detected. So this is one of the advantage, that we don't need to use any probe or specific type of primers for screening purpose. Now expression libraries, they again may be of different types. They may be constructed from eukaryotic cDNA, that is constructed from different type of messenger RNA. And this messenger RNA, again I can repeat that they can represent the total population of the messenger RNA in a particular tissue type. In contrast, the libraries that are constructed for bacteria and yeast, they may composed of genomic DNA, because here the introns, they are not frequent in DNA or sometime in yeast. So that's why genomic libraries, they can be constructed, and their expression product, it can be detected. And for the construction of such libraries, जो target DNA है, इसको हम different methods से digest करते हैं, either mechanical shearing से, या हम partial digestion कर सकते हैं by using different type of restriction enzymes. तो इसका एक disadvantage है जो कि overcome किया जा सकता है. तो जो main disadvantage है वो ये है कि sometime the termination sequences of neighboring genes, they may be present on the target gene as a result of random shearing or partial digestion, and the expression may be inefficient because of the presence of such termination sequences. So it can be avoided if the fragment size of the target gene is controlled so that it is comparatively smaller, and it is inserted into the expression vector in all possible combination or orientation, so that it may be in a reasonable number, such recombinant clones, and the translation may be in all possible reading frames.